And we're very fortunate to have Dr. Kent Bradford, Distinguished Professor and the Director of the Seed Biotechnology Center at UC Davis, and Dr. Megan Hochstrasser, Science Communication Manager at UC Berkeley. And Kent is going to start, and then Megan will bat cleanup. Kent, you have the bios, so I'm not going to go through their bios.
So what I'm going to do is talk a little bit about crops. So I'm a plant guy. We work on improving crop foods, not that animal foods, seafoods, and so on are not important, but that's sort of what we work on. And what I'm going to try to do is give a sort of a setting for next speaker, who's going to talk about the most recent technologies, but give you a little bit of a history of this, and try to get you aware of what we're going to try to do and what we need to do from the plant side, plant-based foods, to meet this challenge that was just discussed. Just over in the aquarium here is a poster on the wall that said we need to increase food production 70% by 2050. So that's what all the people in the agricultural world are thinking of right now. That's going to come very fast. 2050 is not that far away. And as you'll see, that sort of change in productivity has been challenging. So we'll see what we can do. Everything we eat is pretty much has been domesticated. So we eat very few things really in their natural state. So most people may not be aware that this is where corn started from. Teosinte didn't have a cob. The seeds all fell off, and you had to gather them up some other way and so on. And that's what humans started with in Mexico about 3,000 years or so ago, and created corn out of it just by selection. So here we're just talking about selecting. When you think about something like this, that when you shake it, it's going to fall off, some little mutation that allows it to stay on there until you can collect it is automatically selected for. So basically, if you start collecting stuff and then replanting it, you quickly select for things that stay on the plant rather than fall off. Most things are weedy and they want to fall off. So that's a very easy trait to start with, but it's the way we domesticated everything is just what we like, we kept, we pre-planted, and that just comes along with the selection then. And of course, we've done more with that, but the hard work was already done. That is, the humans had already done this before we ever started doing any breeding. And those are based on genetic changes that we'll get back to in a little bit. That's not all we do. For example, we use grafting, a very popular way with all of our horticulture crops, grapes, tree fruits. Every fruit that you eat, every grape that you eat, every wine you drink, and so on is grown on a grafted tree. And if you've ever been by our walnut orchards in California, they all look like this. Why is that? You look puzzled. Why do all the trees look like that? They didn't, they grow that way, but it's because they're on black walnut rootstocks with, English walnuts grafted on the top. Why do we do that? Well, the rootstocks are resistant to disease. They're more tolerant to the soil conditions, but they don't make good, black walnuts are not what we want. We want English walnuts. So you graft them together, and you get what you want. Same with all of our grapes. Those clones of Cabernet or so on that we grow are not on their own roots. Generally, they're on other rootstocks. There we go. The reason I raise this is because these are, in fact, two different species. So we often say, well, you're messing with things, you're mixing species. We're mixing species here, not genetically, but functionally. In other words, we're taking advantage of the characteristics of two different species by grafting them together and letting one do its job in the root and let the other do its job in the shoot. So it combines the benefits of two genetic types. It's very common. Nobody gets upset about it, and it's limited to within a genus or family. In other words, these are both types of walnuts, grapes, both types of grapes. But we do put peach trees on a plum root and so on. So, I mean, it'll go so far, but otherwise they won't work. In other words, they have to be reasonably related or it won't work. Plant breeding is basically genetic modification. So here's Mendel. Mendel worked in the 1850s, but actually his work wasn't rediscovered until about 1902. So it's only a little over 218 years, 216 years that we really first had a grasp about the fact that that heredity really works. That is, you pass on things from parents to offspring. And Mendel's major thing was to say they come in little units. Everybody knew all along that parents' traits end up in their kids. The question was, how does that work? If you think about putting a drop of dye in a jar and you stir it, it just changes the color and it's all diffuse. You can't get it back out. Mendel pointed out that it's not really that way. It's like dropping little beads in there. Each little trait has a little unit, and in fact, they can sort back out in the next generations. Each trait is passed on as a little unit. So that was really quite remarkable, not knowing anything about the mechanism whatsoever, just from the way that the genetics work, that you could track through pedigrees. People had done crosses before. They had crossed plants and grown the progeny and said, wow, that's interesting. Not very many people thought about going to the next generation and the next. They just looked at the first one and said, oh, well, that's interesting. Mendel went to the next generation, kept track of it, and said, oh, these traits keep showing up over and over at a certain ratio. So that's really what started what we'd say breeding, where we realized by systematically crossing certain things, following those traits, selecting those again and again, we can make progress. So most of it is using sexual crosses. Plants are sexual as well. They have male and female parts. They have pollen, which is the male part. They have eggs down inside of the pistil. It's going to form the ovary and so on, and the seeds are the offspring. So if we want to bring in traits from different plants, we make a cross. Again, this generally is only possible with things that are reasonably related to each other. It's very effective. It's rather slow, though, and you can only access certain things. That it has to be possible. Potatoes and tomatoes are closely related, but we can't cross them. They're not related enough that you can make a sexual cross between them. So if they're related enough, we can cross them. But here, you take the two completely different things. It's like taking two baskets of apples and pears. You dump it all into a big thing and round them all up, except there's 20,000 different types that you have to sort out again. In other words, you make a big fruit mix out of all the genes and everything, then you have to sort them out again. And it works, but you make many generations. In other words, the next generation, you can get half of those again, the next generation, half again, so on. So it works, but and it's the main way breeding is done. This is the main way breeding is done, just being patient, making crosses, next generation cross again, and so on. And it's been very effective. So back to the food issue, how effective has it been? This is showing yields of wheat, tons per hectare of wheat, over a long time. So this is the US. And what you can see is that it's quite flat for a long, long time. So a lot of times we hear, well, why don't we just let farmers just, year after year improve their crops and we're going to be fine. It doesn't happen. It just stays flat, okay? Unless you're actively pushing that selection, you just keep the same random stuff going on. About right here, 1960s, yields shot up. That's why we call it the Green Revolution. Maybe you've heard the Green Revolution. That's what it was, is that about 1960, yields started going up. In the US, maybe threefold. Here's India, eightfold increases in some cases. About half of that is from the genetics we're talking about. Half of that is improved varieties, short wheat that would hold up to fertilizer and so on. The other half is things like fertilizer, pest control, management, and so on. So the Green Revolution was both. Big component of it, wheat and rice. Big component was the genetics that enabled these higher yields. And so here we're getting yield increases of about threefold over that period, next 30 years or so. Which was fortunate, because at the same time, the population went from 3 billion in 1960 to where we are now at 7.8 billion. So over that same 30 years, where we increased yields about threefold, the population went from 3 to 6, 7 or so. So Paul Ehrlich, The Population Bomb, was written in 1960 or so, saying it is inevitable, famine is inevitable, forget it, we will never make enough food for those people. It didn't count on the Green Revolution, but it did that. We got to here, the next 2 or 3 billion are going to be harder. So we didn't stop here. It looks like the projections go anywhere from 9.5 to 10.5 or 11 billion before it really plateaus. So the population has definitely increased and is not peaked out yet. The rate of increase is going down. It's slowing down. It's not there yet. The combination of these, though, enabled us to keep, this down here is the per capita production. So that's the amount of food per person, which is fairly flat. We didn't have those massive famines because we didn't have enough food. We've had famines because of bad weather in locations, from disruptions due to wars and all kinds of problems, but it wasn't really because we just ran out of food, which was the scare. This is the production curve. So it increased over that whole period. It's sort of that pace. This is yields, production up there per capita. And the key thing is the area harvested is this one down here. So this threefold or so increase in production did not happen because we put 3 times more land under production. That's on almost the same, maybe a 5 or 10% increase in area on which that happened because we increased the yield per unit area. So that was a huge thing. And what that means, which is really important, I'm going to go back and forth and look at different people once in a while, is that this represents the land area not converted to agriculture due to the increases in yield. Because their study's been done, and they argue that probably we would have taken over that land. That is, we would have mowed those forests down, because when people are hungry, they're going to do it. I mean, they're going to grow stuff there. The fact that we could have this yield increase without taking over a lot more land preserved an enormous amount of land, which also preserved an enormous amount of carbon from going into the atmosphere. One of the ways we put the most carbon into the atmosphere is taking land from its native state and turning it into agriculture, because all that standing vegetation goes up, plowing the soil puts carbon up. So the carbon saved by just not converting that land is about 1/3 as much as the whole period of time since the Industrial Revolution that we put into the air. In other words, we did not put about half again as much carbon as we could have because of this increase in yield. So back to how this, what's the basis of all this? Back to the basis that we're going to get down to tonight, which is that these little genes that, that Mendel discovered that carry traits turn out to be embodied in DNA. So I'm sure you've heard of DNA. It's the molecule that carries genetics and so on. When I was in high school, I did a term paper on DNA. I was a young high school student. I was looking up stuff, and I found out that the Watson and Crick's famous paper describing the structure of this complex molecule was published the week I was born. I'm 65 years old, so 65 and a half years old. April, 3rd week of April, 1953, they published their paper saying this is what DNA looks like. So everything we know about DNA has happened in the last 65 years. You're going to get to where we are now later tonight. But what we do know after, and it took a lot of work, but what we figured out finally was that actually it is the sequence of these little base pairs, the A, T, Gs, and Cs, that encode or spell out the structure of proteins, which then the cell uses to do what it needs to do. So we have A's, T's, G's, and C's, 4 different molecules. And the way they're lined up is just like spelling out a big, long word. You may have 2,000 or 3,000 of these spelling out one individual protein that the body needs or the cell needs. And these are wrapped up into chromosomes, and they live in the nucleus. When the cells divide, they duplicate them and spread them on, and that's the way the heredity works. And then when the cell needs to do something, it takes that DNA, opens it up, makes a copy of it on a molecule called RNA, which goes off into another part of the cell, and it has machinery to make the actual functional things that you need, the proteins you need, all the things that do enzymes, the whole thing runs off of this. So what that means is that if we want to have some specific trait, if we can put it in the DNA, it will come out the other end as some type of trait, whether it's color, flavor, staying on the plant, whatever we wanted. So back to combining species and so on. So here's a domesticated tomato. Don't look at that one. This is a nice big red tomato. Here's related wild species. This is Solanum lecopersicum. This is Solanum peruvianum. They're different species of the same genus, but they're close enough that you can cross them. So in tomatoes, all the tomatoes you eat are benefiting from certain traits that have been moved from these by the breeding process. So all of our processing tomatoes, all of our fresh market tomatoes carry genes, pieces of DNA that have been introduced by crossing, by the sexual process, that enable them to have resistance to diseases, resistance to nematodes, higher flavor, quality, all of these things. So this is just the way it's been done. All of our crops have taken advantage of wild relatives through breeding to bring in improved traits. Here's one, for example. You mentioned delta-ish areas. You mentioned the low-lying areas around the world are going to have a problem. Here's Bangladesh. Very wet place. Flooded all the time. One of my colleagues at Davis, Pam Ronald, and a colleague, David McKill, he's now at Davis, but also worked at the International Rice Research Institute as a rice breeder. They identified a single gene, a single protein in the genome of rice that enables these plants to be submerged totally for over 2 weeks And then when the water goes down, they grew back. So these are the plots here that were planted. All of them were planted. They were submerged for 2 weeks. Take the water off. These mostly died. Even though rice likes to grow with its feet and roots in the water, it doesn't like to be completely submerged like this. All of these plots that have survived and regrown have that single gene. So this shows you the power of just one of those pieces of DNA. A couple thousand base pairs long. Move that over just by crossing. They just moved this by traditional breeding, moved it in. And now this rice is being distributed over there and really saving lives because this flooding is going to happen. And by being able to survive that and then have a decent crop, it's making big changes over there already. So it shows you, again, the power of breeding just by the traditional method is quite powerful. But it demonstrates also what a single gene can do. So I always like to ask this question. White grapefruits, red grapes. How many prefer white if you had a choice? Nobody. How many prefer pink then? The rest of you don't like grapefruits, I guess, at all. The choices are the pink ones in general, though. Okay, we'll come back to that in just a second. Talk about mutations. So mutations, what mutations mean is that somehow a change has been introduced into that sequence of DNA that results in a different trait. In other words, there's an introduced change somehow. The whole process of evolution occurs because of mutation. That is, if you didn't have variation, there's nothing to select on. Darwin says, selection of the fittest. If there's no variation, there's no more fit than any other. Okay, so the fact that continuous generation in biology of variation is a reality. There are mechanisms to generate that. When we want to generate more, we can use ways to induce mutations. We had the Atoms for Peace program, okay, radiation. When it was new, well, we irradiated everything. In fact, there was a giant thing built in Japan with a big berm around it. Bring up some cobalt and radiate trees, and they went after it in a big way. This has been used. It's not that efficient, as we'll get to in a minute, but it does work. Over 2,000 cultivars have been used in induced mutagenesis, just radiation mutagenesis. Several thousand cultivars. In fact, everything you eat has mutations in it because mutations happen all the time. But we have several thousand cultivars with induced mutations where specific things have been, you induce changes, and then you just select from that the ones that have what you want. It's undirected. It may introduce unknown changes and so on. It's sort of a needle in a haystack. In other words, you're making a lot of changes, and then you're trying to find those that have the traits you really want. I'll come back to that in just a second. So back to this. Which one of these is the mutant? The red one. Hate to tell you, but this was originally a natural mutation. There was one tree growing in the Rio Grande Valley of Texas where a grower found a red grapefruit. Before that, they were all white. Who knew? Had a red grapefruit. That was kept and propagated by the grafting mechanism I showed you a while ago. That one tree, that one branch with those branches has been propagated into all of those types. Further mutations were induced in that to make it even redder and make other varieties and so on. When you think about it, what else can we do? How many prefer, say, navel oranges? Anybody like navel oranges? Navel oranges. Why do we like navel oranges? They don't have any seeds, right? They're seedless in the middle. Exactly. So in other words, if we didn't propagate those by cuttings, they're gone. That one was found in Brazil by some missionaries, some monks, basically in Brazil, transported to California 200 years ago, and so on. Robertson navel. So we have cultivars that are continuously propagated because that one, the sexual process is stopped. If there's no seed, you can't go on and so on. So all you have in those is mutation, and then selection, and then propagation vegetatively. Okay, so these are common procedures that are used, and you're familiar with them because you eat them. Now, with the modern techniques we have, we can actually make mutation much more efficient. So there's a process called tilling. Won't go into great detail, but what it is what it says is since we know this process, we can induce mutations in a big population of plants, and then knowing what we know about DNA, we can very efficiently screen tens of thousands of those plants to look for a change in the specific gene we're interested in. So if we know that the change in a particular gene will give us what we want, from white to red, we know this is probably anthocyanin gene. We know what those genes are. we could mutate a lot of things, and using this tilling population, we can then very efficiently screen at the DNA level. In other words, we don't have to grow out 20,000 plants, wait 5 years for them to flower, wait for them to grow fruit, and then say which ones are red. We can go straight to the DNA and say which ones of you have inherited a change in that particular gene, which is the one I want. And we can do that much more efficiently. So we can still use mutation with just selection and just let that process take place. So that's another technique we have because of more recent technology. The other thing that's happened is that the DNA sequencing, which means determining that exact sequence of A, B, A, T, Gs, and Cs. In other words, actually reading that DNA now has become very simple and very cheap. The human genome was done. It cost $3 billion in about a decade to sequence ours. We have about 3 billion of those ATGCs. That's how long the book is, 3 billion of those. It took us about a decade and about $3 billion the first time to do that. They have machines now that can do 60 of those in a day or so. That may be a little exaggeration, but not much. In other words, you can load up machines now that will do that. And it's got so cheap that, in fact, I was at a meeting recently by a seed company, and they said, for us, it's basically infinitely cheap. It's cheaper than doing anything else. We'll just sequence the genome. So we have machines now like these, and we're not to the end point yet. I walked into my colleague's office last year. He had this little thing plugged into his computer. He was sequencing an entire genome on his desk in live time with these ATGCs coming across the screen right in front of us. So that's where we are. So this part is not the problem. We can get that information. So just to say that now it's as if you had this huge encyclopedia, but it cost you billions of dollars to go read it. Now the encyclopedia is there, and we can read it very efficiently. And we can not only, we can Google it. We can say, I want to know what exactly is in there. We can suck it out, if you know what I mean. Okay, so that's what's happening with the technology. So what did we do with that? And if we can do that, one way is to then, if we know the gene we want, we can transfer it by what was called recombinant DNA, which means take a piece of DNA and just move it. So we had a little bacterium, this is for plants, that would do this for us. It did this naturally. This is the way an infected plant has moved a little bit of its own DNA into the plant. Once that was discovered, we said, maybe it could piggyback a gene we want along with it. We took out the pathogen genes and put ours in there. So we just sort of hijacked this little pathogen to be the little vehicle to carry a gene. So if this was the gene we wanted to introduce, now we just cut that piece of DNA. That was discovered in about 1972 in California, that you could cut DNA with little enzymes and put them back together. Cut it, placed it in here, put that in so that it would carry it into the chromosome. So basically what we're doing here is what I wish we had called gene grafting. We called it transforming the plants. That sounds like Frankenstein, and it sounds really scary. Why didn't we call it gene grafting? That sounds more safe, right? And that's what we're doing. Basically, we're grafting this gene just like we're grafting the trees, okay? Except just one gene, not the whole thing, into the chromosome. Once it's in the chromosome, it's inherited, it's expressed, it works. It's like a plug-in. It's like if you ordered a part for a thing and you plugged it in and it works because the mechanism all recognizes everything the same. So that was possible to be done in plants by the early 1980s. We could make these plants, you put it in, grow them back up, and now you've got this plant with just one more gene, the one thing you really wanted it to have. So why do this? Well, the traditional breeding I've described is limited to genes that occur in sexually compatible population. Like I said, if it's in a potato, that doesn't help me for tomato. If it's in pepper, it doesn't help me in tomato. They're cousins. They're very closely related, but they're not close enough that we can do that. So if it doesn't exist within the population we want, we're dead. There may be traits that we want that are in other species that we can move. Even like a pepper, my colleague, we helped move a single disease resistance gene from pepper to tomato. That would enable them to stop spraying lots of copper bactericides in Florida where they grow peppers, where it's very hot and humid and there's a lot of disease. It can't get out. We'll get about that in a minute, but it worked perfectly well. Because it had to be transferred, it had huge regulatory burden. It's still not being used, and in the meantime, they're still spraying copper, by the way, to protect those plants. So we can introduce novel traits now that may not exist anywhere that if we wanted to do some other things. For example, we can make medical products, proteins, all the insulin that anybody who's diabetic used is produced by this type of genetic engineering. Put the insulin gene into a yeast or a bacterium, grow it up, harvest the protein, that's where it all comes from. And specifically, we try to enable environmentally beneficial practices because, again, if we're going to increase productivity by 70%, we can't at the same time destroy the environment by 150%. We really have to do both at the same time. So what were the key first targets? This is just showing, just look at all the crops. Insects, diseases, and weeds, losses. Third of the total productivity can be lost to these three things. Okay, so obviously those were the first targets that people looked at. How can I stop wasting 30% of our effort to things like weeds, diseases, and insects? Lost productivity means less food or more land used. That is, if we lose a third here, we need another third land additionally grown to get that product. Okay, so this is just a trade-off we have to deal with. So one of the traits was herbicide resistance. What this is showing is planting corn right into where they had corn before. They didn't plow the soil, till it up. They just left this stuff on there. Unfortunately, these green things are weeds. If you don't plow, you got weeds. Okay. What they did is they made the crop resistant to a herbicide, weed killer. So you plant the crop, let it come up, spray the whole thing once with a weed killer. It kills the weeds and not the crop. You don't have to plow. You don't have to. You leave that mulch on there to stop soil erosion and so on. So this trait right here, which people vilify, it's Roundup Ready, it's Monsanto, it's all these things, actually has done a huge amount to increase conservation tillage, reduce soil erosion, reduce the size of the dead area in the Gulf of Mexico coming out of the Mississippi River because it's not carrying nearly so much soil. So that was just a single trait that has had a huge effect. Pest resistance. Caterpillars like this eat a lot of crops. They can be killed by a particular bacillus, a bacterium, Bacillus thuringiensis, that infects these, produces a protein that stops their gut from working well. They either quit eating or they ruptures their gut and they die. It's called BT. It's used in organic production. It's totally safe for humans because we don't have that little receptor. It doesn't hurt us whatsoever. So this protein, the DNA encoding that protein was taken, putting into the plant, so that now this is corn that is expressing this one single protein. The insects can't eat it. Same for cotton. So now we have a lot of corn, a lot of cotton that is expressing this BT. It doesn't have to use pesticide. So here's when it was introduced in cotton, 1996. Cotton is one of the highest pesticide-using crops we have. This is a bollworm. If you don't control the bollworm, you don't get any cotton. It's just that easy. You can see once they introduced this BT cotton, the pesticide use went way down. In fact, the combination of this control and intensive monitoring and pesticide use, we've pretty much eradicated this in certain areas of cotton production. Now we don't need to use any pesticides for that particular insect because we had this in combination. It's also a lot safer for workers. I mean, here, this is maybe a way a person applies pesticides. A lot of it is sprayed on. If you go around the world, this is the way they apply pesticides. Mix it by hand, pouring it out, walking right in the spray, and so on. It's not safe. A lot of these pesticides, at the level they're at in the final product, are not harmful. We monitor all the food in California, fruits, vegetables, everything. They're always tested. Used properly, it's very, very low in the final food. But if you're a worker, this is where you're exposed to the pure stuff. It's very careful. I did that when I was a kid. I can guarantee you I got sick. I didn't handle it properly, I guess, or something. Because so pesticides, we may need them, but we'd rather not. We'd rather have some way that we can build into the plant to be resistant. Just like you'd rather, I hope, have a vaccine than try to cure smallpox or something after you've got it. Right? It's much better to have a flu shot than it is to try to cure the flu when you've got it, because we have very little that'll do that. A few other things we've done, don't want to run too long. Virus resistance crops. We don't really have any pesticide or any way to control viruses in plants. We can try to stop the insects that transmit them. We can do various things, but we don't really have any way to do that. This is ring spot virus on papaya. By 1995, papaya in Hawaii had pretty much been wiped out because of this virus. I mean, this is pretty unsellable. The plants are really sick. Pretty much a dead end. Researcher at Cornell, Dennis Gonsalves, used some genetic engineering. He enabled the plants to recognize this virus and counteract it. So basically, he made a virus for the plant. I mean, a vaccine for the plants. Plants don't have a blood system where you can just make an injection and so on, but he made them basically the same way. Recognize the virus, stop it, and be immune to it. So now all the papayas pretty much you get from Hawaii, they're not organic, they're rainbow variety and so on. Those are the result of this one trait. It's been working for 20 years, working perfectly well. There are movements continuously in Hawaii to try to get rid of it. People don't like it. I guarantee you if they take those out, this will be all they have left. The reason they can grow organic papayas in Hawaii is because the virus has been taken care of by this mechanism. Okay. A few new things on the market. You may have heard of the Arctic apple. Who may have heard of the Arctic apple yet? Okay, it's about to come out. It's an apple that when you cut it, it doesn't turn brown. So basically, it's one gene, one single enzyme that operates and makes this brown color. They just turn that off, and now it doesn't make the brown color. So, studies show that kids are more likely to eat this than they are that. Studies show people would rather eat sliced apples than whole apples. It's too much. It's hard on your teeth. I'd have to cut them when I eat them. So this is on the market now. They're making little dried chips. It's slow to build up. So there's very little of it. It's been legalized. They're building up, apple orchards and so on. It'll take 5 years or so. But keep an eye out for it because it's the other thing they mentioned is when you make juice, what color do you think of when you think of apple juice? Brown, brown, brown. You know why? Because this enzyme is functioning. When you crush all those apples, that enzyme is functioning, it turns it brown. It turns out when you make juice out of these, it looks like Chardonnay. Maybe it's more attractive. Maybe people say, well, I like my brown orange apple juice. Fine, you can probably have that. But you might also like it looking nice and clean like a Cabernet and still be apple juice. So they're putting this into multiple varieties, again, using just a little bit of technology, silence one single gene. Potatoes, there's a so-called an innate potato, reduced bruising. This, when they bang around and so on, it causes darkening and they waste all of that. It also reduces browning upon frying. In other words, everybody likes their French fries to be crispy but fairly white. Nobody wants them to turn really dark brown and so on, so that will stop that. And it also reduced the acrylamide production. We're in California, right? How many of you have seen the little sign in Starbucks, right, where you put the milk in your coffee? Where it tells you and has to inform you that roasting coffee produces acrylamide. Has anybody ever seen that sign? It's always there. We don't look at it anymore, but it's true. If you fry or you roast or something at high temperatures, certain things in certain plant materials will form acrylamide, which is not good for you. It's low quantities. It's not going to kill you probably at drinking coffee, but why not get rid of it? So what they did is they made these so when you make French fries, you don't have acrylamide in it. So it's a consumer benefit as well. So let's look at the global impacts of these technologies. This is a survey from the first 15 years or so. 10% reduction in pesticides. A lot of people will tell you that GMOs mean pesticides. They don't. They've reduced total pesticide use since they've been introduced. Reduction in environmental impact. Again, because I said less plowing, less carbon in the air, those types of things. Less fuel used. Again, less plowing, less spraying, and so on. Less greenhouse gas emissions. Again, same reasons. Another more recent study, which included particularly international, particularly the cotton, and this is only from cotton really in India and China that have been commercialized around the world. 37% reduction in pesticide use, 22% increase in crop yields, 68% increase in farmer profits, particularly in developing countries. This has been a game changer for like cotton in India, places like this, where those people are sending their kids to school. The women are not working 80 hours a week weeding. These types of things are changing because of this technology. So they have been the big winners. Nobody tells you about that. They tell you it's all the companies making all the money. The big winners have been the farmers in these countries. And this guy's done some excellent studies, full-time studies starting before, medium, after introduction, longitudinal studies that are very, very, very thorough. A few things that are not out there yet. Golden rice. A lot of very poor children around the world who pretty much can eat only rice. That's basically what their families can afford. They are going to be vitamin A deficient. 1 to 2 million of deaths of children a year are occurring from vitamin A deficiency. Okay? So there was a professor in Germany who said, I think I can fix that. So they found 2 genes, one from maize and one from bacteria. 2 particular genes in a pathway could make beta-carotene, which is the precursor of vitamin A, make golden rice. Anybody heard of golden rice? A few of you heard of golden rice. So that would do it. In other words, this has enough rice. A reasonable portion of that would satisfy the minimum daily requirement of rice. This is 2013. Protesters, led by certain anti-GMO groups, invaded International Rice Research Institute and tore up all the plots that they're having to do in order to meet regulatory requirements to release this stuff. So they've done everything they can to delay that. So it's still not on the market. We're 20 years down the road nearly from the first report, 14 years down the road from when it was certainly good enough to give them enough vitamin A, still not saving children. So this is the controversy that we'll probably talk about, but it is not out there yet. There are other projects. We've mentioned Africa. Critical things, same thing. It's like golden corn. Why not? There's golden sweet potatoes, orange sweet potatoes, which is not their normal thing. They eat white sweet potatoes. But they're working very hard to show people if you eat orange sweet potatoes, your kids don't go blind, your kids don't die. I think that's pretty easy sales to make, really. So there's a lot of work going on, drought tolerance, a lot of different things. A little bit of this technology. All it means is a few genes a lot of times that will make this big difference. Further along, here's some work from one of my colleagues at Davis. Sodium chloride, salt. That flooding in Bangladesh I mentioned, that's not nice fresh water. That's salt water coming in on those lands. Central Cali in California, salty land. Okay, salting of land is a huge problem. Salinity is going to be limiting our land. One gene that he found in a different species, put into the tomato. These are the controls growing on salt water. This is the ones that he got one gene change, made it quite tolerant to salt. So these are the types of changes that we could potentially do to enable our crops to grow better under these conditions. Same guy, Eduardo Bloomwald, same lab, drought tolerance. These are the control plants. He droughted them for 15 days, no water at all. These were the controls. They never recovered. These had the same treatment. Then he watered them. 7 days later, they came back. So basically made resurrection plants out of them almost. Huge value to this because if you're going rain-fed land, you're going to get alternations of times when it gets a little bit too long between rains. If they don't just give up, the plants just hold what they got and wait for the rain to come again, we have a chance. Now, not 100% yield, but say 60% rather than 0. That's huge in a lot of areas. Another colleague at Davis, disease resistance. I won't go into the technology, but basically, we can now identify the specific organism that is the pathogen and engineer the plant to recognize it and stop it. No pesticides, nothing in the environment, no off-target effects to anything else. That specific organism, the plant says, you won't grow, I'll stop you, and it goes on. Okay, so these are the disease ones, these are the controls. So this is possible technology now as well. The nice thing we have now is this new technology, CRISPR. This is gene editing. This is where actually, I mentioned this big encyclopedia. Not only can we read it, now we're going to be able to go in and make these changes, letter by letter, and do the things instead of hoping it happened or using these a little bit more tough approaches. And Dr. Hochstetter is going to talk about this to you in great detail. So I'm going to finish here and say, why do we want to use this technology? It extends a biological method, crop genetic improvement, that's been remarkably successful in increasing crop productivity and quality. So I tried to give you an example. This is the method that we domesticated crops with. It's the same method that we're using in breeding. It's really the same thing. We just know more about how to handle it now. It can reduce pesticide use. It can help conserve soils. It can be more sustainable than what we're doing. It solves problems for which we have no other solutions. I mentioned plant viruses. There's some of these we just don't have other choices that we could go with this. It can improve nutritional content and health. We have a project in Africa where we're breeding for higher nutritional content. Why not? If every bite you're going to eat, why not have more nutrients in it? Right? This is not that hard to do. Now, we can make food production more efficient so that land can be preserved for wildlife and biodiversity and so on. Edward Wilson, one of the most renowned ecologists we have, basically says if we want to preserve biodiversity, let's go to the places that are really biodiverse and leave them alone. In other words, that won't happen if the people around there have to encroach on that more and more to grow food, because that will happen. People will do it. So the key thing is to keep our footprint of agriculture the same, increase productivity, reduce losses, and try to meet this need of increasing food production without increasing the scope and area of land. So with that, I'm going to hopefully set the stage for the really interesting and exciting technology and answer any questions you have.